Hello, everyone. Thank you for joining today. During today's talk, attendee will be muted. If you have any question at any point during the presentation, you may submit it at the chat panel. At the end of the talk, there will be a Q&A session. And if you'd like to ask your question yourself, let us know to unmute you. And as a reminder, and remember, there is a dedicated Slack channel for this talk on the Brain Space Initiative workspace. And as a reminder, the presentation is being recorded and will be shared publicly. It is my great pleasure to introduce to you a distinguished scholar and a researcher, Dr. Dean Salisbury. Dr. Salisbury is a renowned expert in the field of psychiatry with a specific focus on psychosis and auditory neurophysiology. Dr. Salisbury's research helped change the understanding of psychosis and pioneered the use of combined brain imaging and EEG to demonstrate the link between gray matter loss and auditory impairment. In 2012, he joined the faculty at the University of Pittsburgh School of Medicine Western Psychiatric Hospital to continue his research on identifying biomarker for early identification of the disorder. Dr. Salisbury has been a prolific writer, publishing extensively in the top-tier scientific journal and receiving numerous awards and grants for his contribution to the field. Dr. Salisbury, thank you for being here today. And without further ado, I will let you take it from here.
Well, thank you very much for inviting me and for that gracious introduction. So let me see if I could share my screen here. How does that?
Perfect.
Okay, great. And all right, so thank you very much again. And what I'd like to do is to go through, and I just want to lay out what we want to do is to give a little background on schizophrenia and psychosis, to talk about some of the earlier work of lesion studies combining EEG and MRI, some basic postmortem studies that came up with this idea of dendrotoxicity in the disorder, and then talk about distributed circuit function or circuitopathy that we get at a little bit using spectral connectivity measures. And then along the way, what I wanted to do was to try to indicate some needs for better tools. And I just want to sort of have a disclaimer that the tools are probably out there in the engineering and neuroimaging world, but in psychiatry and myself, we may not be aware of them and everything. So I think that could be a place where it'd be really sort of fruitful for us. So, we study people with schizophrenia, and schizophrenia is a disintegration of the different facets of personality, particularly between the mental and emotional spheres. And so schizophrenia means split mind. It doesn't mean, like multiple personalities or anything like that. It means split mind. And so what we're looking at here, in about 1% of the population with a typical onset in early adulthood, it's very, very debilitating. Over half of the people, in young adulthood or late adolescence will never be able to work at all for the rest of their lives. So the disorder is characterized by several positive symptoms that are generally not present, such as hearing voices or seeing visions, paranoia, and irrational beliefs or delusions like conspiracies or strange powers, delusions of reference where things relate to you, or bizarre delusions like aliens and things like that. There's a cognitive aspect of disorganized thinking and speech, which is a big part of it. It's also characterized by several negative symptoms, a lack of things that are generally present, such as anhedonia or a lack of pleasure, attention problems, working memory problems, and those are subserved a little bit under the cognitive domain too. Perseveration, getting stuck on ideas and things and thinking. Avolition, so inability to initiate behaviors. And apathy, which is really just sort of a lack of caring in some sense, but really sort of engaging and being motivated. And we talk about this positive and negative. Sometimes the negative is broken down into enduring deficit or more phasic or transient non-deficit. And there are other ways to break it up, but really positive and negative and cognitive are the ways we sort of generally think about the disorder. So just to give you an idea about what we're talking about, this is from an interview we did with of someone who'd been ill for a long time, and voices are a major symptom. And about 70% of people with schizophrenia experience auditory hallucinations, even when they first get sick, when they first are in their emerging psychosis. About over 70% will hear voices. And so this was an interview, and we said, well, when you say you got sick, you mean like hearing voices? Yes, hearing the voices, yeah. And now you said the voices scare you. Is it because you're afraid they might make you do something you don't want to do? Yeah, they say the whole time you're going to do something bad. That's what the voices say. Okay, but otherwise, except for the voices, you feel safe. Yeah. How often do you hear them? Like, do you say right now you're hearing them? Yeah. Are they constant? Yeah, pretty much. And sometimes they get real bad. How many voices do you hear? Many. And do the voices talk to each other? Yeah, they do, as a matter of fact. And that's sort of like a next level of illness when the voices talk to one another and not necessarily to the person. And do they keep a running commentary on what you're doing or thinking? Do they comment on your behavior? Well, they just read my behavior that I'm scared, and then one of them would go like, "We got this, kid." So it's typical to have a very negative sort of connotation to the voices of someone with schizophrenia. Usually they say bad things about them. Okay. All right. Is it male voices or female voices? Both. And do you recognize any of the voices? Yeah, I do. Is it people you know? People I knew. And why do you think the voices are there? I mean, what's your understanding of it? I believe it's judgment. So judgment, you think it's like evil spirits or something? Yes. All right. So this would be pretty typical from one of the individuals that we might see with schizophrenia, where voices are a big problem. And it's one of the reasons why we focus on the auditory system, and because the temporal lobes are where auditory processing occurs, but also in the left side in particular is where verbal language is located. So it's one of the reasons why we focus on simple auditory processing in the disorder, because there's a disorder physiologically in that hearing and language system. In the end of the 1800s, 19th century, Emil Kraepelin really sort of put together a lot of different psychiatric disorders and called it dementia praecox, or precocious dementia, which became known later as schizophrenia. And we sort of think of him as really trying to get at the biology of schizophrenia. So he said these considerations force us straight to the assumption that we're concerned here with a palpable pathological process in the brain. We thus come to the conclusion that in dementia praecox, partial damage to or destruction of cells in the cerebral cortex must probably occur, which mostly brings in its wake a singular permanent impairment of inner life. And so we've known for over 100 years that there's probably structural abnormalities in the brains of people with schizophrenia, but we weren't really able to find them. So although changes were searched for over 120 years, the only reliable structural deficit until the 1980s was enlarged ventricles. And there were various ways to look at that. Pneumoencephalography took the cerebral spinal fluid out and filled it with air. And this was occurring in the early 20th century. And then they took serial x-rays, and you can see that the ventricles were larger in people with schizophrenia, but you can imagine how invasive draining the CSF and filling it up with air is. And CAT scans weren't quite good enough to get at the subtle pathology. I mean, we could see that the ventricles were large, but we couldn't necessarily get at the cortex. And then MRI came around in the 1980s. And so when we even talk about postmortems, until, you know, the 1980s, 1990s, it was really considered that schizophrenia was the graveyard of neuropathologists because there aren't classical signs of neural degeneration in the disorder. It's very subtle, and we just did not have the technological advances to be able to detect that. And so it really wasn't until the 1990s where we had advances in MR methods, especially the millimeter voxels or volume pixels, finally gave the spatial resolution necessary to detect the subtle changes in schizophrenia. So in addition to the MR, we've used EEG, and EEG gives us a high temporal resolution measure of brain function. But as you can see, of course, it's recorded from the scalp, and so we're looking at the envelope of volume conducted activity that we can get. The advantage over fMRI, which would be in the brain, is that EEG and Meg, which I'll talk about later, are more directly linked to neural activity, but they also have a millisecond temporal resolution that's just unmatched in other imaging modalities. So real quickly, for EEG, we need at least 2 electrodes to record it, a reference, or what you might call an inactive electrode. That's not really true, but a reference electrode, and then something closer to the active source. And what we're looking at is the activity in the superficial layers of the cortex and the dendrites in the pyramidal cells in the cortex. And what we do is we look at a potential difference between 2 areas. So when we have electric current flowing, right, it's the right-hand rule that generates a magnetic field. And so when we have activity in a large group of neurons, a population of neurons, we can record the electrical and magnetic fields that are generated in the population of neurons. And so if we think about it at what scale we're looking at, here we would see synapses, which we could look at synaptic currents, we could look at spikes. At a mesoscopic or mid-level scale, we look at LFPs, local field potentials. And then in the EEG, we're really looking at the macroscopic scale, okay? And there are maybe, you know, 100,000 to 1 million neurons that we're recording from and seeing it in these EEG waves here. This is a depth electrode, and what we have is a surface recording. This is a cat visual cortex while they're being shown visual stimuli down here. You can see the activity in the multi-contact electrode shows a polarity inversion around layer 4, which is where you'd expect it to be. And as we get to the superficial layers, we can see that the recorded surface EEG looks very much like the superficial activity in the cat. And that's pretty much true for humans. And so what we're looking at really is the LFPs that are making it up to the superficial layers, and we're recording it in the EEG. So, all right. So when we think about schizophrenia, I'm going to talk about a lot of the work that I did. So it's going to be a little bit myopic in that sense. So hopefully you'll indulge me. And if I leave anybody else out from another thing, it's only because we want to talk about what our lab's doing, and we don't mean to make any omissions there. But Marty Shenton and Brian O'Donnell were working in Bob McCarley's lab at Harvard when I was there as a postdoc in 1990. I joined the lab. And Marty had brought MRI into the psychiatric clinic, and Brian was a neurophysiologist as well. And this is one of the first studies in schizophrenia combining MRI and EEG. And what we found, this is the radiologic convention, so this is the left side of the head, is that there was selective volume loss in very ill patients in the left temporal lobe more than the right temporal lobe. And that the P300, which is a particular brain wave related to auditory target detection in this case, was reduced in patients. And the amount of gray matter loss they had on the left was associated with reductions in P300. Okay, the left superior temporal gyrus contains a lot of the auditory signal generators, And it also was correlated with their thought disorder. So one of the symptoms, so the smaller the volume, the more disordered their thought was. So this was one of the first studies, but it took a lesion or region of interest approach. And so we went in and measured those MRIs slice by slice, just like that, and the computer reconstructed them. Now, at the same time, there were also Voxel-based studies, or a little bit later, that were going in to try and look. And this is a very interesting one at UCLA from Ty Cannon. He's at Yale now. But what they looked at were monozygotic and dizygotic twins, and those that were discordant for schizophrenia. So what you had was, this is plotting the correlation in gray matter volumes between individuals. So here we have healthy monozygotes. And what red means is that they have a high correlation between the twins, very similar cortical volumes in a voxel-based method, okay? Healthy dizygotic twins, who are really just siblings, have much less of a correlation, right? So this is the genetic contribution to the volume here. When we look at monozygotes, though, that are discordant for schizophrenia, what we see is that there are still areas that are highly correlated between individuals, but the temporal lobe and the frontal lobes show less of a correlation than you would expect in monozygotes. And so our approach, measuring areas, showed temporal lobe abnormalities. This VBM one showed frontal abnormalities, pretty widespread frontal abnormalities, left and right, but also temporal lobe abnormalities, and actually some visual cortex here. But these are really the two culprits that come up, frontal lobe and temporal lobe. So what happens in our first episode patients? Well, our first episode patients, in '92, I went to McLean, and Marty and I set up a first episode laboratory there as part of a program project grant. And Marty handled the MRI side of things, and I handled the EEG side of things. And we were testing people within 1 year of their first hospitalization for psychosis. McLean Hospital is a psychiatric hospital in the Boston area that pretty much gets people from all around that New England area. And we were testing people there. So we went in and we looked just like before, and we wanted to see what's going on in the auditory cortex at first episode. Yoshio Hirayasu was there, who came over from Japan and slice by slice measured the superior temporal gyrus and that superior temporal plane there. And the computer reconstructed Heschel's gyrus and planum temporale. And so just to point out, these are anatomically defined, obviously, right, by sulcal gyral patterns. We reconstructed them. And what we found was that there was moderate bilateral reduction in Heschel's gyrus, and that contains primary auditory cortex. And then the planum temporale, which is secondary and tertiary auditory cortex, a little bit more posterior, usually has a leftward asymmetry in a healthy individual. And the idea is Geschwin's hypothesis that has to do with language processing. There was marked left hemisphere volume reduction in the first episodes there. So even at first episode, there were reductions in that auditory cortex that were more marked on the left. So to bring it back into the neurophys part, Yoshio had also brought over another measure called mismatch negativity. And basically, it's just a very simple brainwave that's elicited by a change in sound. So it just goes beep, 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 boop. And if that boop or that change is salient enough or big enough, you'll orient to it. But that's an automatic process, right? It happens outside the focus of your attention. And it's generated in the auditory cortex, particularly in the early sensory auditory cortex. And what we showed was that, well, actually, in first episodes, that response was pretty healthy at the group level. So this is a negative brainwave, about 100 milliseconds or so. And here's healthies in first episodes. It's pretty healthy. They're a little tiny bit reduced, but not really markedly. However, when we looked at the MRI of that left Heschl's gyrus volume, okay, and the mismatch, what we saw was there was a correlation in our patients where if they had a very small mismatch, like an abnormally small mismatch, they had reductions in the left Heschl's gyrus volume, but not in the right, okay, only in the left. So this was selective to the left hemisphere. There was no relationship in healthy controls between the size of their mismatch and their volumes, or in people that had affective psychosis, mostly people with, mania. Here we are, I had that backwards. Here's our healthy controls here. So it was really only the people in the schizophrenia spectrum where we saw some people that had really bad mismatches and very much reduced Heschl's gyrus volume. So it's important to notice we're looking at scalp activity that we assume is coming from Heschl's gyrus, which is a relatively large or gross anatomically defined area. Well, we followed these individuals over time, about 1.5 years. Kyoto Kasai, who's the chair of psychiatry at University of Tokyo now, we saw that the left, but not the right, showed progressive reduction in cortical gray matter. And when we looked at their mismatch, same individuals over time, obviously a small n, we saw there was a reduction over time in the same individuals in the schizophrenia spectrum, whereas our healthy individuals and our people with affective psychosis didn't show reductions. So we're seeing a progressive change in that mismatch response. We're seeing a reduction of gray matter on the left, but not right, auditory cortices. And it's highly correlated. So the worse your mismatch gets, the more volume you lose. And even if you don't necessarily believe in the correlation, I think it's important to notice that all of the patients, for the most part, in the schizophrenia spectrum are in that negative quadrant, except this one fella. Okay? There's no relation between change and mismatch in the other groups. Okay. So there is some sort of progression going on. And so I think this is really one of the more important studies that came out. This came out in 2017 from Australia. And basically, this is not a longitudinal study. It's just a large study of individuals cross-sectionally, but they're broken up by 5 years' time from their illness, okay? And so you have people from 20 to 25, 25 to 30, 30 to 35 as we go down. And what you'll notice when we look at the gray matter is the temporal lobes and the frontal lobes, temporal lobes and frontal lobes, are hit first, okay? And as we get to closer to 30, a little longer duration, we still have more widespread temporal and frontal, but you can see it's spreading, okay? And as we go longer and longer in the disease course, it's spreading to the rest of the brain. By 35, 40, we see widespread whole brain gray matter loss, okay? So we were looking for these lesions, say, it's temporal lobe, you're going to find a lesion in temporal lobe, or frontal lobe, you're going to find a lesion in frontal lobe that leads to this disorder. But probably what's happening is that early in the disease, it's initially focal, right? But later in the disease, it becomes diffuse, and it becomes one that's affecting the whole entire cortex and other parts of the brain, probably subcortically as well. And so what's going on to lead to this loss of gray matter? And I think most people now in the field would agree that there's some sort of a dendrotoxicity or a loss of dendrites. This is some work from the chair of my department, David Lewis's group. But what you see here is a healthy pyramidal dendrite here with synaptic vesicles and dendrites on it. Sorry, it's an apical dendrite. And here you see in the person with schizophrenia and another person with schizophrenia in postmortems. They've been ill for a long time. But you don't really need to count. You can pretty much see how many dendritic spines that they've lost on their cortical pyramidal neurons. And this is true in the prefrontal cortex, which is a particular area that Dr. Lewis is interested in, but also with Rob Sweet at Pitt has looked in the auditory cortex, and again, this is in the spine density. And what you can see is that there are much fewer spines in persons with schizophrenia in area 41. Brodmann's area 41 is roughly speaking Heschel's gyrus, and 42, roughly speaking, is planum temporale. You can see that the patients have much fewer dendritic spines, and so what we have, is a disorder of dendritic spines, a disorder of loss of connectivity between neurons. And it initially starts in frontal and temporal areas, but then it spreads to the entire brain. And part of it then is as you're losing connectivity between frontal and temporal areas, you're starting to get into a distributed circuit abnormality, not a focal lesion abnormality. So again, just to highlight that, what we see in people who have not actually had psychosis, this is called the clinical high risk for psychosis group. This is a group out of Australia. And again, it was the left temporal areas, without getting into too many of the details, but these are the people who converted. It was the left temporal areas that were predictive of who was going to transition to psychosis if they lost continued volume there over time. Again, from Ty Cannon at UCLA, I think he's probably at Yale by 2015. But anyway, again, they're finding the frontal areas that were predictive of conversion. So again, temporal frontal. And then this is a large Enigma study that just came out on clinical high risk individuals. You can see there's almost 1,400 healthy individuals, 1,200 people that were clinical high risk that did not convert to psychosis. And then roughly speaking, 18% who did convert to psychosis, 16, anyway, less than 20% who did, 18% who converted to psychosis. And so, again, one of the three major areas that was predictive of conversion was loss of cortical thickness in the temporal lobes. Okay, so a lot of that was just to justify why we were looking in those areas. And so back at Pitt now, we ran a new sample of first episode individuals. Now these individuals were within about 2 months of 3 months of their first clinical contact for psychosis, and not all of them were hospitalized. So unlike our older studies where it was within a year of their first hospitalization, this is within 3 months of their first clinical contact for psychosis. And Anna Schaefer at the time went through and manually traced Heschel's gyrus again. Okay, so we reconstructed that here. Heschel's gyrus, this is a semi-inflated brain here from Free Surfer. And we replicated that correlation between volume and mismatch. Okay, the mismatch, more negative in this case, is better because it's a negative brainwave. Recorded from the scalp again. And also duration, so just different kinds of mismatches. So we replicated that. But the issue is, you can't, it takes forever to go through, right? And measure a gyrus, right? So we really want a automated parcellation scheme. And the question that, we need engineers and imagers and radiologists and is to answer is what's the best automated parcellation scheme, that's fast and accurate, maybe has subcortical components to it, and that can account for pathology. And what I mean is when you do the warping into a template or a template into the brain, that you can account for the fact that these people may have volume loss in certain areas. And so these are all the things that need to be addressed. So real quickly, one that's often used is the Desiccant or the Desiccant atlas. It has 35 ROI, and the thing is, they're anatomically defined on sulcal and gyral patterns, right? So if we run this, we can get superior temporal gyrus, but it's really gross, right? It's just a huge measure. It's not terribly useful because there are many different areas involved in here, right? Different functional areas involved in here. So this doesn't really get us past an ROI where we can go in and pick things out. But this is really very commonly used The Destro Atlas is better. It has 58 parcellation units. And we can pick out in here, that little red line in there, Heschel's gyrus, but the rest of the STG is sort of all merged together. And so again, it's anatomically defined, maybe not the best thing. So what we've been applying in the lab is the human connectome Glasser parcellation. And what that does is it uses multimodal imaging to get a high resolution automatic parcellation that's really sort of what I say is quasi-functional. And so the first thing is it does is a myelin map, right? So a T2 weighted, white matter weighted map, obviously structural maps, task related fMRI, and then functional connectivity maps. And one thing you can do is make a myelin gradient, right? And so, you know, sensory areas like auditory cortex down here, when you have a sensory cortex, you have a lot of input into layer 4, more cortical white matter there. And so that can be a clue as to breaking up functional subunits. And then your gradients here all put together in a multi-shell model, and you end up with a very parcellated, quasi-functionally parcellated map of the brain, 180 areas per hemisphere. This is a flattened one. This is from David Van Essen's part of this project. And this, if you've, well, you probably weren't born in the 80s, but anybody who was in grad school in the 80s in the visual system, Van Essen was making these flat maps. But what this does is it gives us a really nice functional rendering of the auditory cortex. And so, excuse me, we were one of the first groups, if not the first group, to apply this to early psychosis. And Mark was a graduate student at the time. Here we did it with a relatively low contrast T1 image, but you know, we have been doing it more recently with T1, T2, resting state fMRI, everything you need for that. But what we did was we could now parcellate and say, hey, this is A1. This is primary auditory cortex, okay? And this is one of the belts that's anterior to it. And this together is Heschel's gyrus. And so we validated that we could accurately break it up into auditory cortex functional areas. And so what Mark did first was to take the mismatch we recorded from the scalp, again, just scalp mismatch, apply it to the HCP parcellation and look for correlations. Remember, we had been going in and manually tracing that red area, Heschel's gyrus. Now we broke it down. And what we're plotting here is the correlation between inferior frontal gyral areas and auditory cortex areas and mismatch reduction in our first episodes. And we could see for pitch, we're getting A1 in some of the belt areas around in the auditory cortex that correlate with reductions of scalp mismatch. And for duration in particular, that right inferior frontal gyrus has a nice strong correlation, as well as widespread area activity in the left. Right inferior frontal gyrus is sort of the auditory frontal executive, among other things. So we showed that we could use the HCP and get reasonable results and parcellate things into functional units. But what we really want to do is to get into brain space. And so to do that, you know, we need to record the EEG or the MEG and project it back into a realistic head model. And you may or may not be aware of the problems associated with that, but you know, you have a limited number of sensors. You want to put it into a lot more places in the brain, and so the source solution is not necessarily unique. And so, for example, one thing we can do is just make a dipole of auditory activity. This is for an N100, and here's the N100. You can see a butterfly plot of it. And we can fit the dipoles with very little residual variance, okay? Less than 1% of residual variance. So it's accounting for the scalp variance pretty well. But the problem with a dipole, of course, is it's sort of circular. You have to put it in a region. You have to sort of restrict it moving around because they like to move down to the neck. That's mathematically a better solution. And you have to make sure that they move together. So there, you know, there's a little circularity involved. But our dipoles end up about where we want them. But when we look to a distributed or a Clara in this case, which is which is an iterative kind of source solution. You can see how much dispersion we see. It's really not terribly useful. And it's much better than Loretta. You see a lot of Loretta papers, but the dispersion there is huge, right? So we don't have really great methods that are dipole based, as far as I'm aware, for getting at the activity. And so this is from Ben Hay, who recently came to Pitt, to CMU, sorry, right next door to Pitt. And you have dipole models or distributed models, and the distributed models are sort of where we've gone. But you can see how many different classes that you've got, and from I'm not a radiologist or an engineer, and I told Armin when he asked, I said, well, I'm probably going to be the worst mathematician in the room, So my thing is trying to understand a little bit about it, but to find the best tool. So that's what we need, is to know what's the best tool, what's going to have the least dispersion, what's going to be the most accurate, and how do we apply that. So for me, it's a tool, and that's where we need things that are developed that can help us. So what we did was we went from EEG and we brought in Meg. And what we're doing is we're using combined EEG. So these are very flat electrodes here that are in the cap, and then the Meg, which is the big doer here, that has squids or superconducting quantum interference devices that can measure the magnetic field in a shielded room. And so we get the EEG and the MEG simultaneously from our individuals. And just real quickly, we talked about the measured electric fields that come out from the currents that are flowing extracellularly in the apical dendrites here, the superficial dendrites. And then the Meg field is going to be the right-hand rule. So you put your thumb in the direction, right, and you curl your fingers around, and that's the flux that's occurring. And so we can record those somewhat perpendicular, okay? And so each of these methods is going to have some similar information and some unique information. And sort of the EEG is better for gyral sources, and the MEG is better for sulcal sources, but that's a really crude rule of thumb. Obviously, we can record things from big areas like the superior temporal plane here in EEG. We just have to be at the right spot. We're not going to see much here, but we will see a lot more here, and vice versa with the magnetic field. So what do we do? So what we do is we get a realistic MR. And you can see we've reconstructed it here, but that's actually not an MR. That's just a bunch of points, okay, that are vertices. So that's actually a tessellated surface of an individual MRI. And we're using a distributed source reconstruction or a forward model. And so what we're predicting is, based on all of these little dots, which are vertices, what would the electric field, the magnetic field look like, either or, at the surface of the scalp from those little tiny dipoles, thousands and thousands of dipoles in there. And we use the minimum norm estimate, or MNE, which reduces the amount of energy across the cortical surface in the solution. It's very widely used, and it gives us good results, but we're not entirely sure if it's the best method, So we get our EEG or MEG. Here it's an MEG helmet with the squids. We represent in space where those are. We get a realistic brain. We project the activity back. There's a slightly exploded brain here for sulci and gyri. And then we map the parcellation scheme onto that to see where the activity is, okay? And so just if you're interested more specifically what our pipeline is, we do our EEG and MEG processing. We have our structural MRI. We make, in this case, boundary element model surfaces. We process the EEG and the MEG. In this case, we're looking at a particular event-related potential that we want to project back to the cortical surface activity using minimum norm estimates. So again, one thing that we do need in the field is a method that merges EEG and MEG. And again, as I mentioned earlier, there should be common and unique information, but the current canned methods don't provide better solutions. They seem to be dominated by the EEG. And so there's a need there for that. We really need to figure out what's the best way to get this information used, because presumably you reduce dispersion and you increase the accuracy of the solution. There have been studies that show that, but it isn't widely done, and it's sort of a need in the field. Okay, so I'm going to talk now about some more interesting, maybe, brain space studies that we did using those methods I just talked about. And so one thing we did was we did a sort of a Luck-Vogel task. The individual is getting a cue to direct their attention to the left or the right to remember what they see. They see an array of 1 or 3 objects. So the high load is 3, the low load is 1. And then in this case, they would have to remember these guys. And after a period of time, they have to indicate if any of them changed. Okay, and they have to keep that memory online for a full second. Okay, so they have to retain that memory for a second. And you can see in this case, there was a change from that blue one, and they should say, yes, there was a change. And what we're doing now is we're plotting alpha and gamma activity in the visual cortex here. Okay, so what we know from animal literature is that when you're attending to something, you should see a decrease, in this case blue, a decrease of alpha activity, and there's an anti-correlation with gamma activity in the sensory cortex, and the gamma activity probably reflects local sensory processing in this case. And so what we see in our first episode individuals compared to our healthy individuals is when they're attending left or right with a high load, our healthy individuals show an increase in gamma and a decrease in alpha in the contralateral auditory cortex. That's not there in our patients, okay? So this was sort of the first way that we were getting at that source activity localized to the visual cortex. Alfie Sklar came along and followed up on that when Brian had done the original analyses. And what we looked at was we knew now that we had these seeds in the occipital areas that were showing this abnormal alpha response to load. And then we used phase locking value across the entire brain, okay, to see what other areas were modulated by load, okay. And then we looked at the PLV modulation within the networks between groups. And so we built the network in our healthy individuals. And then we wanted to see what's going on in a widespread network in our first episode individuals. And we could see that in the blue, we're seeing, green is load modulated connectivity with left hemisphere, and red is with the right hemisphere seed, okay? So we know that the middle frontal gyrus here has to do with working memory and spatial visual attention, and that was modulated very well. But we can see basically that our first episodes really didn't modulate to the extent that our healthy controls did in our PLV. And so this was sort of our first step at trying to get at a distributed thing using phase locking value. So the phase modulation between the two areas, how much more in phase or less in phase they became. Another thing we looked at to try and get at this was the N100. And this is actually, sorry, the M100, the Meg version of that N100 I showed the dipole solution for. This is to an auditory stimulus, about 100 milliseconds or so. This is a butterfly plot of the Meg. And this is probably the signal of the auditory percept that is available for perception. They call it stimulus or sensory registration. But in our healthy controls, when you're ignoring a tone, you get a certain sized M100 or N100. And then when you attend to that tone, you see an increase in the size of it. So you could just see, broadly speaking, it's larger. Patients have a smaller M100 and N100, and they don't modulate as well. When we project that back to the cortex, it's really quite nice. In our left auditory cortex, we see discrete activity in A1 and parabelt. On the left, you see widespread activity in the right sensory areas. These are just the reflections, we believe, of the unfolded cortex. So we're really sort of focused on here. And then when you attend, you can see we get hotter colors, right, in these areas. So we're getting more activity in these areas as a function of attention. And attention has to be from a frontal lobe signal, okay? Our patients just aren't showing a good sensory response in the first place, and they're not modulating it with attention, right? So this tells us there is a sensory abnormality, but in addition to the sensory abnormality, there is a cognitive abnormality or attention-related connectivity with the frontal lobe abnormality. So I want to get into how we got to that because I know we're running a little bit out of time. So what we did here was, as part of Mark's dissertation, he looked at all the areas that showed an increase in activity during that M100 time. And there were many areas, including the precuneus, which is a medial area that's related to attention. There were frontal areas, obviously, more on the left than the right, temporal areas, and insular areas that were related to attention. So it's pretty widespread. So how do we figure out how these things communicate with one another? And so what we did was, first thing we had to do was we had to say, well, what seems to be the frequency that's driving the auditory attention effect? And we used phase amplitude coupling, okay? Phase of the low frequency carrier signal and the amplitude of gamma. And what we found was that it seemed like theta, unlike the visual system, which was alpha, in the auditory system, it seemed like theta was the one driving that. And it was significant in all the auditory, it was present in all the auditory areas, but because of multiple comparisons, it was significant in the left A1 only, although the same pattern was pretty widespread. So now we sort of identified, this theta activity as our carrier frequency. So we went in and using the cuneus as a seed, okay, we did all-to-all to the entire vertices of the rest of the brain from all the subdivisions, and we plotted out how many different areas seem to be related to cuneus increases during the attention, okay? So what we're doing is we're looking at each each vertex in that area and seeing how it maps to all the other vertices in the brain, and then reducing the data as a function of the HCP parcel. And what we found was that for the left hemisphere, there was a significant increase in connectivity in a left hemisphere auditory and slightly frontal area. And then for the right, there were frontal, area 46, and actually some visual areas that seemed to increase. So I'm going to skip over this and finish up with this. And so basically, those were really seed region-based things we were trying to get at. And so how do we just let the data drive it, okay? One thing we did was we did a quantitative MEG analysis of continuous resting MEG. So it's just like fMRI, but we're doing it in spectral space. And we're using this vertex-based all-to-all phase locking value. So the first thing you do is collapse across all vertices. And what we see in our schizophrenia spectrum psychotic individuals is they have a reduction in alpha across the brain. And this is not just alpha power, it's the coherence or the connectivity between vertices. And so alpha, for whatever reason, seems to be less coherent in our first episode schizophrenia spectrum individuals. And when we look in the vertices to see where they are, we get 3 clusters that have different all-to-all connectivity patterns. in the brain. And so we could say, hey, these are the clusters that are really driving this group difference, 1, 2, and 3. Okay, you see the precuneus is part of that. And then these are the areas that are functionally connected to it. Okay, and so that's all to all, but you can imagine it's not very powerful. A different way we approached that was using network analysis, machine learning. And so this was with engineering. Henry was a undergraduate, actually, and Irvin was the professor in engineering. And they did non-negative matrix factorization to look at that alpha abnormality. Here, we sampled from, again, over 20,000 nodes, 10,000 plus per hemisphere. Downsampled it, in this case, to Brodmann areas, which are cytoarchitectonically defined. Did a PCA to find the alpha component in here. And then, again, did a functional connectivity matrix. And what we came out with were 4 areas, okay, that were reasonably reduced in the first episode individuals. And so it was pretty much anterior and posterior cingulate cortex that had problems in connectivity. The left auditory cortex, which is, of course, near and dear to my heart. And then widespread right inferior frontal gyrus abnormalities, and then actually some of that posterior parietal abnormalities that popped out. And so this was an application here of that method. So, okay, so we're going to skip through all of this. I'm just going to say thank you. We almost got to everything. And then we can have some time for questions. I saw there was some questions.